Thank you, everybody, for being here. Actually, as Mario pointed very well, my idea of this seminar, although it is multi-omics technologies and approach towards neurodegenerative disease, I have made one specific sections to see what is the advance that they are already out there or that they are on the way. And I'm going to use the first part of my presentation to expand a little bit about that and the systems biology, which is the topic of analysis, all these technologies. But the other two parts of my presentations, I'm going to use it to present some of different omics examples using the works that I have been involved in, and in all of them I have been leading the projects. And the whole idea is most to give you an overview and like to show it in an easy way how these projects can be done and how everybody that from seeing patients or having animal samples or even cell cultures, you can develop these kind of projects and do some kind of proteomics or metabolomics or even integration analysis without being super expert. I just have to comment I first touched a computer in 2000 and it has been 20 years and now I am working in trying to develop interactomics pipelines, but I am not the expert. I just surrounded with the people that are very knowledgeable, and I just transmit what I want from them. Then the whole idea of this seminar that I have prepared is to give you like this kind of same input of we can do all these omics technologies in collaborations with people that is expert, or we can even outsource some of the heavy lifting work, and we can do some collaboration steps, and that's the whole idea of this seminar. Okay, I am going to start just to give you a brief idea of what is systems biology. I was talking about system biology. Basically, is a holistic way of analyze different biologic systems, and is kind of a cycle. In which you can feed with initial omics data or start with public available databases to start analyzing this data, establish some models through different creation of networks analysis. And the outcome of this analysis is going to render the generation of new hypotheses. And this cycle needs to be completed because then you have to go further and test the hypothesis and put that information back and retest the system in order to try to, every time of iteration, get a major proximity of what is happening in reality. Okay, then I am big fan of system biology. I started learning about this like 4 years ago. And it shows basically the difference between what was believed as the central dogma, that it is one gene, and the consequence of the mutation of that gene is going to have a genotype. Nowadays, with the system biology, there are multiple genes, and there is different kind of networks and interactions between genes and regulators of these genes that are going to produce different kind of phenotypes, and some of them are going to be responsible for the disease. Then it's not a straight line, it's a multifactorial components that is responsible for the phenotypic response, and in this case, the disease. Here, I just have a representation of 4 major components of life, of cells. We have DNA, RNA, proteins, and metabolites, including all the lipids in there. Basically, metabolites are small molecules, more than 1,000 kilodaltons. DNA and is the information that have all the code to produce all the proteins, and the final outcomes are the metabolites of all these. Then thanks to the advance in the technologies, we are able to characterize the profile of all these molecules, and then we can see the genome through genomics, the transcriptome to transcriptomics, metabolomics, and proteomics. But they are more than that. It's even more complex because, for example, modifications in DNA and proteins that assemble the DNA, they can call epigenomics, or nowadays we can even do the sequencing of single cells within a complex tissue and have the transcriptomics at the single cell level, which give us a lot of information. And thanks to the recopilation of many informations, we can have different kind of interactions that can be done in silico. Then we have the interactomics. And the metabolites, since they are products of enzymes, they can be actively measured in a time-dependent manner, and we can have fluxomics. But there are other kind of information that can be generated between the interaction of all of them, and this kind of information is going to develop networks or gene regulatory networks that are like a higher level of complexity of information. Then it's just to give you an overview of all the different type of information. And respect to the neurodegenerative disease, and I'm going to concentrate mostly in Alzheimer's disease, is something that I am getting into a study. I'm getting into as a part of my development in my career as an assistant professor. Is like I'm starting to develop some projects in Alzheimer's disease, and I have been collaborating with different groups in other projects. Is known for Alzheimer's disease that at the times that the disease is diagnosed, when the symptoms show up, there has been already several years in which the several molecular markers are already present in the brain or in the tissues or even in the fluid. And so far, they are already specific characteristics of the, for example, in the cerebrospinal fluid, amyloid beta protein is reduced and increase in the tau and phospho-tau in this fluid. Although this is a difficult kind of substrate to obtain and to measure, and there are several studies that has been developed in order to identify markers in the blood or in other more accessible fluids like saliva. And there are other different image technologies from the brain, like MRI or PET analysis, that can also has been developed to characterize the brain in Alzheimer's disease, and they has been currently used in research and in diagnosis of Alzheimer's disease. Here we have the image of a controlled normal brain in respect of the hypoacquisition of the 2-fluorodeoxyglucose that is radio-labeled and do not get into the cerebral frontotemporal cortex compared to the control. And this is a proof, it's a Pittsburgh binding molecule that is able to interact with the amyloid plaques. And these tools are able to characterize the functional and the change that occur during Alzheimer's disease. I just want to emphasize that the major risk for dementia for Alzheimer's disease is the age, and there has been some other like metabolic changes that can be associated with the development of Alzheimer's disease. And I am very interested in characterize or apply metabolomics and proteomics to try to identify how these patients that have just change in the glucose levels, even at early age, they can develop Alzheimer's disease, or they have increased the risk of to have Alzheimer's disease, similar to what happened with the people that have, or the patients that have diabetes, that increase the risk of having Alzheimer's disease. Then the systems biology approach, basically what is telling us is that we need to study each of the components of at the cellular level within the tissue, in the brain in this case, and also at the different fluids, including blood, saliva, cerebrospinal fluid, to characterize around what is known for the major characteristic of this disease, or this neurodegenerative disease. And basically is applying all these technologies, omics technologies, that are going to be able to characterize in a global manner and in a more comprehensive way what is happening in reality in these patients. And I would have to say that it's a hard task. They are already advanced and there are a lot of works that are in progress. But even harder is to try to have a meaningful information by combining all these data, because this is a huge task, and there is a requirement of a lot of bioinformatics tools and support. Then there are some advances that are in progress already for Alzheimer's disease, and there are some targets that has been identified. It's one of the major function of resistant biology is to identify biomarkers that can allow us to follow the disease and evaluate the possible treatments. Some of them are well known, and some of them are already using in clinical, but most of them are still in research. And I just want to make a parenthesis here to highlight why it's so difficult to have a biomarker out there in the available for the patients. There are too many ways or too many possibilities for a biomarker can be not good for the clinical use at the identification step, like there is a lack in the study design or during the execution of the project, or there is also like a lack of budget. I'm going to show next on the slide, or during the confirmation at the end, there is not a good planification for the experiments and the results are not significant. And the identification of biomarker is a long process. It's not that long as a drug development, but one of the things that required for all that process is funding. And one of the things that the biomarker projects lack is the protection of the intellectual property. By protecting the possible biomarkers that are identified in different projects, you are securing that like the entities that have the resources, like industry, can promote the development of these biomarkers. Then the idea is that the system approach will offer new opportunities for developing diagnosis for neurodegenerative disease by analyzing and doing implementation of these multi-omics technologies at different levels within brain tissues or samples from different body fluids. Then this will have the promise of the output of this will be possible biomarkers that have to be tested in the populations, in large populations, and validated. And the whole idea is that this signature of genes will be able to classify a different kind of neurodegenerative disease. That's what we are aiming with this approach. And just to give you schematic representations for multi-omics approaches, we can have two kind of approaches in which we start from the genome first, and we identify one mutation, and then we scale up what is happening at the epigenome, transcriptome, proteome, metabolome, or even microbiome level. Or in the other one, there is a characteristics, let's say, for example, in the case of Alzheimer's disease, we have the accumulation of the amyloid proteins, and then we can start around there going backwards or around then to see what is happening. And there, I'm going to give some examples of them. And this is just a schematic representation of a model in obesity in which different kind of omics technology identify different kind of genes. In this case, in yellow are the genes that have several mutations that are responsible for the risk factor of obesity. And this is at the GWAS analysis level. But when there is a pull down of a specific mutations, and then they were able to identify through the chip sequencing analysis genes that are nearby or distant that are regulated by this initial gene. Then this is another layer, the epigenome. And when they do a transcriptome analysis on this kind of samples, then they were able to identify a whole different profile of the gene expression of the genes that are associated with the risk factor to become obese. Then when they identify these mutations in these genes to be relevant, they test these few mutations by CRISPR technology, and they found which of the mutations actually resemble the profile of genes transcription. All these kind of analysis required to be confirmed and validated in vitro assays, also in doing the mutations, inducing the mutations or the deletion of the gene, and in specific mouse models to resemble actually some physiopathology of the disease. And this is the genome approach. And what has been lately advanced during the last couple of years at the GWAS analysis for Alzheimer's disease and Parkinson's disease is that there is now the possibility of super large studies in which there are millions of individuals that has been analyzed for millions of SNPs, and they identify a significant numbers of these genetics association mutations. And here I just highlight how some of the pathways that has been clustered for these genes for Alzheimer's disease, in which we have here the amyloid metabolism, AB amyloid precursor protein, and A beta aggregates. And we have that the ApoE4 is like the major risk factor gene, even in different pathways. But we have also immune inflammatory response pathways, lipid and cholesterol metabolism pathways, and other basic cellular process that basically what they have identified so far mainly are like mutations that are protecting or against the development of Alzheimer's disease. Then this is one advance at the genome level, but in reality, what is needed is to start doing this multi-omics analysis in different systems, and each of the cells that are in the brains that are implicated, or the functional assays, need to be evaluated in order to get this large amount of data and integrated with this genomics data. In this is like an editorial where they are questioning the use of multi-omics technologies for Alzheimer's disease. Basically, they say that the lack of all the clinical trials efficacy or the drugs that are in development for the COPD is because they has been going around only into around the amyloid plaques theory, and a holistic and multi-omics integrative analysis will be more significant to be able to classify different type of dementia and differentiate it from normal subjects. And these have to be in the context of aging. And that's something that have to be in consideration and need to be addressed towards that end. Then I just want to explain shortly, I mentioned before, like gene regulatory networks are kind of a high level of interaction, because one gene, that is transcribed, and finally the protein that is produced can regulate the expression of another gene, or can to be expressed, or itself can control the expression of this same protein, or can inhibit some other proteins. Then the expression of only one gene finally ended up affecting distal genes that have nothing to do directly by them, but it's the effect of someone else. This creates some networks, and in order to develop these networks, there is cellular information that need to be acquired at the genomics, transcriptomics, and proteomics level. And once you acquire this data, you can start generating this kind of interactions or maps. And in order to do that, the data have to be processed, have to be clear out and develop some algorithms, test these different algorithms with an initial subset of data and validate it in a large set of data in order to be able to have like a model that can give a biological significance. Then all of this is to explain, all this explanation of these gene regulatory networks is to tell you that there is some work that is already going on. I bring this paper that is not peer reviewed yet. It was on this biorepository posted last month. And basically, they are start to doing this kind of regulatory network analysis for independent cells. We have excitatory neurons and microglia, and we have also in the context of Alzheimer's disease and schizophrenia. And you can see here, like basically they are able to map across all the chromosomes what are the genes that are interacting or can regulate distal genes in this kind of interaction. This is like a visual way to see all these interactions. And we have that information, or that information is coming up from excitatory neurons and microglia and oligodendrocytes and inhibitory neurons. Then, and this is the kind of information that is going to actually fill up all the system biology together with the proteomics, metabolomics, and different kind of integrations that are going to be generated with using clinical imaging data or functional analysis. All of that need to be processed, and there are different tools that need to be generated, and some of these pipelines are already out there for other kind of disease. Like in cancer, for example, there is a huge advance in the integration of multi-omics technologies for biomarker discovery. And the outcome of that is going to be identification of novel pathways or networks that can be now target for drug development or for use of biomarkers discovery. And one of the major characteristics is that all that information is going to allow to classify an individual subject if have predisposition for the disease or have the disease using all that information, which is going to resemble the phenotype. Then that's one part. And in the case of neurodegenerative disease, there is an extra layer of complexity that can be incorporated to the analysis of this data. And that one is the neuroimaging, in which there is like not only structural imaging, but also functional imaging. And they are already different type of algorithms that has been developed to be able to classify like different subjects from subjects that have Alzheimer's disease, as I find out in this example, in which there are different subtypes of Alzheimer's disease at the structural level or at the functional level based on this neuroimaging analysis. Then for neurodegenerative disease, we can find, or there is under development, a lot of omics that are going to interact with all these brain image analysis, and that is going to give like a better view, a more integrated view, and I think it's very promising for the development of actually interventions that can prevent the progression of susceptible subjects. Then there is like a new topic, and this was developed at the Institute for System Biology in Seattle, in which basically we have here the system biology, system medicine in one arm, but that one have to interact with the healthcare system, all the online information, all the electronic information about the subject, and the involvement of the patient itself to be able to predict the development of the disease in a very personalized manner. in a preventive way in which the patient or the individual is actively participating to prevent the disease. Let's say, for example, in my case, I am from a family that is diabetic. I am in my 40s and I start feeling the losing of my memory. I better start taking all the precautions. If I do a genetic predisposition, more likely I can find one of the genetics markers, and I need to then get active. What they are proposing is to have like a support, like a virtual support from all health care system to promote like healthy behaviors and to do follow-up on these kind of patients. Then I think at the end, this will be something like the system biology and the multi-omics technology will integrate with the medicine data and medicine systems to be able to provide this kind of good outcomes for subjects in a different way. On the other hand, there is the availability to develop drug development for using all that information. I'm going to start giving you the examples of the omics technologies that I have been working on. Then I have like 3 examples, one for proteomics, one for metabolomics, and one for omics integration analysis that I have been working on. And one commonality for proteomics and metabolomics is the mass spectrometry, which is the way to acquire the masses of these molecules. And they require a separation system, which can be HPLC or UPLC, and the detection system that is the mass spectrometer detector. At the end, you get a profile of this spectrum. Each of these peaks have hundreds of data acquired that belongs to one initial molecular weight and the fragmentation fingerprint of each of them that is used to identify the molecules can be according to the molecular weight when the information is crossed with a database. In the case of proteins, then there are different type of proteomics approach that can be used. We have a global, which is unbiased. Basically, you don't know anything. You know only the samples that you are processing, and that belongs to a specific disease. In this case, let's say Alzheimer's disease, brain tissue, compared to brain tissue from the same area of people that was otherwise cognitively unchanged. And then you compare, you process all that and identify the profile of that. You can have identified thousands of proteins. Nowadays, you can identify around 15,000 out of the 25,000 genes. But the low throughput is low, meaning that require a lot of time of analysis per sample, and give you a lot of profiles of these proteins. In the other hand, we have the specific or targeted approach in which you develop specific methods for proteins of your interest, and it works. It is as a specific as an antibody, but it's antibody-free. And you're required to know, have a lot of information of your protein, and it's a high throughput. While this can take 6 hours to run, you can run this in 10 minutes per sample. Something like that. This is one example of one of my projects, and COPD project that I have been working already like for 10 years, and during the last 5 years I have been founded on this project. And this was the starting point in which we compare goal zero or healthy, otherwise healthy, but subjects that smoke the whole life against goal 4 that are the very severe. And there was a big band here. We identified the unbiased way more than 2,000 proteins on that extracting from these gels, and we identified histone 3 to be increased by 8-fold. We performed a post-translational modification analysis and identified that in COPD, this histone is hyperacetylated, and this was confirmed using different immunoassays. The goal for the COPD patients that have increased amount of the histone 3 and the acetylated forms of the end term. Then the acetylated version of the histone 3 can discriminate all the COPD patients from all the controls, which was important. When we characterized the presence of the histone 3, we found it extracellular. by immunohistochemistry. We found it in the bronchoalveolar lavage, is the liquid lining in the mucus of the lungs and in the plasma. And it's known that when the histones are extracellular, they are toxic. By this annexin 5 assay, we can see that histones induce apoptosis in a dose-dependent manner. Histone 3 is the more toxic of the histones, and even the histones from COPD can slightly more toxic. If we overexpress the histone 3, there is a little bit of toxicity, like 10%. But if we overexpress a protein that is secreted, is highly toxic. It can reduce the viability by 70%. And the toxicity of histone 3 can be quenched by specific antibodies against the histone 3 and or C-term, which make the histone 3 as a new target for drug development. All that was published like 5 years ago. And during these last 5 years, we have been able to advance. And just lately, we identified the specific source of the accumulation of histones using high content imaging analysis, which is a high throughput confocal. And we are able to quantify per cell the content of the acetylated histone in the lung of the COPD subjects. And we were able to replicate in vitro by exposure to cigarette smoke the accumulation of the acetylated histones, and even we are able to detect the acetylated histone in the accumulation in the cytoplasm of these cells. We use CRISPR technology to put like a fluorescent tag, red fluorescent tag, into the endogenous histone 3, and we test the expression of that protein in the presence of cigarette smoke, HDAC inhibitor, and proteasome inhibitor, because we found, in addition to the acetylation, we found ubiquitination in the C-term of the proteins. And what we found is that the combination of all the three is able to induce the acetylation of histones, as we found in the lung tissue of the COPD subjects. Then, in order to prove causality, we take these control cells and the ones that accumulate histones and induce apoptosis, and put it in top of primary human alveolar epithelial cells. And basically, over the first 12 hours, we can see change that these apoptotic cells that are enriched on acetylated histones induce the damage to the primary lung cells. These are the type 2 pneumocyte cells that are the same as the target cells for the coronavirus that has been already characterized. Then we have developed these in vitro toxicity assays or in vitro models for characterizing the disease. And we are using these in vitro models to do drug screening. In this case, using the same high throughput phenotypic screening in our confocal, we are able to have a positive control with a molecule that prevents the toxicity and a negative control in which all the cells die. And we are starting to have positive signals from a kinase library that can protect from the cell death in the presence of extracellular histones. Then that is an example of how a global approach, like starting for the phenotypic, the accumulation of the histone, we can go and extract like what are the pathways or what are the different implications, and how we can use this information to do drug development. We have also developed a method to the quantification of this protein in the plasma, and it's also a target for biomarker use for COPD. Then in systems biology, the way to generate knowledge is have a control system, like a cell model. In this case, is a representation or it's an example of this paper with a yeast in which they have overexpressed or knock out a specific enzyme and another protein that regulate the production of these enzymes. Then after that, they start doing each of the different omics technologies. And then that way you can feed the system and start getting some hypotheses to corroborate in vitro. Then the following examples are like another proteomics assay in which we have induced the overexpression of a protein. In this case, in the context of neuroaids, it's known that HIV infect and kill lymphocyte, but it's not well known or that famous that they also infect macrophage and survive. And actually, the macrophage, they go and they hidden into the brain, and they can live longer there, and that's one of the major source of they work as a reservoir, and they can reappear when later on after several years of treatment. Then, in order to understand better how this function, we characterize this BPR protein overexpressing in a system, and in this case we have another technology in which the cells are labeled with heavy amino acids, and we process the samples at one, and we can tell the difference in only running one sample, and this can be corroborated later on. Then during this analysis, we identified the profile of proteins, and these are the list of the 40 most differentiated proteins. And when we perform a pathway analysis, several pathways during the glucose metabolisms were identified, and they were as a major component of the proteins that were classified there in this pathway analysis. When we corroborate these proteins, and we found that several of them were in the glycolysis, pentose phosphate, and citrate cycle pathways. Then these proteins were differentially expressed, and we identified a possible way in which the BPR is controlling the metabolism of the cells in order to promote the new synthesis of viral particles and the long survival of these cells. Then with that part we conclude the proteomics, but I will return, I will continue now with metabolomics. And in metabolomics we can have the same as in proteomics, like a global metabolomics. There are more than 3,000 different metabolites in the body, and these get even large by every time we eat food, and we get like hundreds of more metabolites from the food. Then sample preparations for metabolite analysis is even simpler than the proteomics. They basically, you can have a lysate and just do all the precipitation of the proteins using methanol or chloroform, and then the metabolites can be just concentrated and analyzed by different technologies. One of them can be quantitative, and the other one is just like a qualitative. In this one, you can get the identification of the specific methodology of the specific compounds depending on the methodology. And in this one, it has been required some training and identification of where this compound is within the running of there. But in general terms, the message that I want to give here is that you can process your own samples and send it to analyze to third-party companies. These company, Human Metabolome, for example, they charge you like something around 500 per sample, and you can have like up to 600 different metabolites that they can identify. And you have a good idea of the metabolites, or you can also concentrate in specific pathways, in this case, like central carbon metabolism, including the pathways that I found that were modified. And they basically have area under the curve that is measured, and they just have some mass spec points to identify that belongs to that specific metabolism. Then coming back to the hypothesis that was generated through the proteomics work, we aim to develop a target metabolomic analysis to see what happened with the products and the substrates of these enzymes in the same context. And we developed these flux omics, starting with heavy glucose, and we were able to measure the incorporation of the heavy glucose and the breakdown of the glucose in all these byproducts through different pathways, including glycolysis, TCA cycle, and even measure the excretion of the metabolites that are coming from the glucose metabolism. In order to do that, we have to do, for each of the compounds, have to develop a mass spec and have to be develop a standard curve to test the sensibility. And here we just have an example. This is the parent ion of the heavy glucose and the 2-fluorodeoxyglucose and the two daughters or fragmentation ions that are measured right there. Using this technology, we were able to characterize inside of the macrophage what is happening when we overexpress that protein in a time-dependent manner, 1, 3, and 7 days. And basically, we found increase in the glucose uptake, but different change in the metabolite production and accumulation of a specific metabolites. In this case, you see that this protein is overexpressed, and this metabolites is accumulated. Although this protein is increased there, that doesn't necessarily mean that it's functional. Then what we found is that by this being not functional, is repressing, and all the system is leading to the production of glutamine and glutamate, and the glutamate can be released extracellular, and we found the same molecules, alpha-ketoglutarate, glutamate, and glutamine extracellular, and this can have a consequence in the exocytosis toxicity of the neurons. Then this was evaluated independently. This is basically what I mentioned, 7 days using these cell lines, and they were also evaluated in primary macrophage. After 14 days of, we were able to detect increase in the glucose uptake and increase in the extracellular release of glutamate. Then these are two examples in which we have used proteomics and metabolomics in the same project, and they can be complementary. But doing the integration of all this omics technology, it can be complicated. It can be really challenging, and there are different steps. One of the major complications is to the normalization of the different source of data. They are measuring different things, or they can have different values. It's required a lot of ways to being able to do this kind of integration. And I'm going to present now a project that we are developing with my PhD student. She's finishing already this project, and she's going to defend in a couple of weeks. Basically, the project is about drug repurposing using multi-omics integration analysis in COPD. And the drug repurposing is basically find a new use for a drug that is already out there in the market. That is going to save a lot of time and money for drug development. And there are several examples of this drug repurposing approach using systems biology. But there are some other famous, like the use of Viagra that was identified used by a retrospective clinical experience. Then what we need is to apply more systematic approach for drug repurposing, which can be advantageous. Then basically, in order to have a very strong drug repurposing identification, there is also a combination of different omics approach. We have genetics, pathways, analysis, and this can be combined with phenotypic screening or binding assays in order to be able to identify these new possible targets in a specific disease. And I just want to mention that we have spent a couple of years setting up the infrastructure and the softwares that are required for this type of analysis. And these are some of the outcomes that we have been able to identify. We some of the data we have been analyzed ourselves, like this GWAS analysis for COPD, in which we identify by integrating 36 different analyses, 73 genes that are related to COPD. And we can find a specific signature of genes that here in yellow are the controls that are overexpressed only in the COPD patients. And these genes can classify in upregulated proteins and downregulated genes that are differentially expressed from this transcriptome analysis. And doing a pathway analysis, we can pretty much ensure that the immune response is playing a major factor in this subset of patients that we are analyzing, that are the very severe COPD patients. And by incorporating genome, transcriptome, proteome, and metabolome analysis, we basically try to see what are the commonalities between them, and we then found major overlapping genes. And then we decide to incorporate a protein-protein interaction analysis from the transcriptome and proteome outcomes, and we were able to actually have a better way to integrate all these different omics analysis, and we were able to identify 153 signature genes, of which some of them have genomics implication or has been related to mutations in the COPD analysis. Then this is basically the pipeline that we have developed using different sources of data. And the last step is to evaluate which of these signature genes can be druggable, meaning that has been related to a drug effect or can be altered by different drugs that are already in these two different databases, drug bank and druggable genome. Then the outcome of what we have so far is that we identified, starting from 16,000 genes that were analyzed, we identified 81 within the signature gene of the COPD that can be already the target for drugs, and some of them are in clinical trials or in development, and two of them have the genetic component of the COPD, and they can be modified by these two drugs. In the case of lansoprazole and plerixafor, both of these drugs are in clinical trials for COPD. And then we just to show you another application of omics technologies. This CXCR is a chemokine receptor, and we try to identify using single cell sequencing databases where inside of the whole cells of the lungs are expressed, and we found that they are expressed basically in all the immune cells, from macrophage, natural killer cells, B lymphocytes, plasma cells, dendritic cells. They are highly expressed there. Then we are confident by these results. We need to do now confirmations using tissues, samples, and publish that data. Then I just want to say that as to finish, as I said at the beginning, I am interested in apply this omics technology into the Alzheimer's disease. I want to just present what are basically my major aims. I am currently developing cell models to characterize like a phenotypic way of the disease, in which I am looking for different ways to increase the amyloid formation in primary neuronal cells, and use diabetic models that are known to have cognitive impairment, and to characterize specific segments of the brains like prefrontal cortex and the hippocampus and characterized at the metabolomics level and proteomics level in order to start elucidating some pathways that are related or that are altered in response to the high glucose content that resembles, for example, in the case of the COPD patients. Then this is kind of the project that I am currently developing in my lab with all the resources. And I just want to thank all the support that I have received from different members within the university, outside the university, in my lab, that have like 3 years already old. And Fang Wang is the PhD student, which have done a lot of the integration analysis, and she's graduating, or she's defending this month. Okay, then I have been funded by the Flight Attendant Medical Research Institute for the 5 years, and I have also funding from Temple University. And I just want to thank you for listening, and also thanks the support that I have received from my family in order to do all this research that I really enjoy. I am happy to have any questions. Thank you.
Excellent. Thank you very much, Dr. Barrero. Wonderful presentation. Yeah, so let's open it up to any questions. Again, you can unmute and ask your question, or if you want to use the chat, that's fine as well.
Dr. Barrero, muchas gracias. So
My pleasure.
This was an amazing presentation. Really, I think that I have a lot of questions. There's a lot of information, but I first have to get out my main feeling right now, which is that it's amazing, the integration, and we had many speakers talking about multi-omics, but I think that the vision, the end vision of making this really soon available as a drug or as a target for modification, it's very unique. And I think that your ability to integrate, and particularly to propose hypotheses to be tested with integration, it's extremely valuable. So we are seeing more and more developments in the technology side that it seems like really go very fast. But many of our Alzheimer patients and people in the world come from a very challenging context, social context. And one question that occurred to me when you were talking about this is where the social context. We've heard about epigenomics and all that, but really personalized medicine and have to do with not only the individual person, but the context. So how do you integrate both life course perspective and also the different social and behavioral contexts?
Okay, thank you for the comments, and that's a really nice question, and it's challenging by itself only do the integration of the omics technology, and you saw the layers. Like omics is already very complex. Now you put imaging, and it's a totally different platform even for the computational people that is in charge of that. And now what you are saying is like you have to put like let's say like the demographic data of the subject, of the patient.
And or experiences like trauma, for example.
Yeah, that's all the history of the patient. And I can tell you here at Temple, we have like the computer science technology school. They has been interacting with the hospital, and they are developing some algorithms to involve all these kind of data that are basically inside of the history of the patient. Okay, then they are trying to incorporate they are trying to develop this kind of algorithms, and also, for example, like behavioral components of the students, for example, in another kind of work, because they are able to track the ID card, and they basically use the card for everything, to get in, out of the room, the gym, the lunch, then tracking all these new technologies. For example, with this device, you can also put all your information, how many steps you did per day, and if you feel also how well are you eating or how much are you eating, then all this information, it can be integrated. It will require like these superpower servers, but more importantly, like the algorithms that are currently developed. And what I'm trying to show here, like even though I have been working in diabetes and HIV and COPD, all these tools can be applied now for this specific topic that I am interested. Then the same here, it is the other way around, is it no matter what, if you want to try to do this kind of corporations, you just need to find like the right people that can help you or guide you on how to start doing this kind of analysis. But there is what I'm trying to, the message here is there is no limitation. If you have some kind of data that you think that will be good to analyze, like there is already, like what I was showing you, like the more than 1 million data of patients that has been genomically classified for mutations. Then you can try to evaluate if that kind of data is in that kind of population, and try to do new kind of analysis on them. But it's totally doable, and I think that's the future that will be, in order to be able to develop something that really change this kind of pathologies that do not have any treatment. We need to start identifying it at the early stage and change the behavior of all of us in order to be able to have like a healthy aging.
Thank you.
Thank you.
I thank you very much, Dr. Barrero. This is a fantastic presentation. I really enjoyed it, and it's very complicated, you know. So with this, you know, very highly dynamic system, I guess what you are measuring, right? I mean, all of this, you know, omics. How would you like separate, you know, the state from the trait, you know? I don't know if that's an issue that you dealt with.
How I separate this?
Like state from trait, for example, some psychiatric disorder, people could go in and out of episode, you know, and even with the question of Dr. Maestre about the social, you know, influence, which I would imagine it would reflect in some omic changes as well, right?
Yeah, I think that's a really nice question and very challenging. I will say that, like, for example, let's say like exacerbation episodes, they can be different, like factors that trigger that episode, and that by itself is one of the one area to study, like what happened in the same subject when he is, let's say, in a control stage versus during an exacerbation, or like let's say an episode of schizophrenia. Yeah. Then it will basically, the patients and the family know, for example, like drug or alcohol abuse in a psychiatric patient will trigger the episode, or for example, change in the let's say in the diet can also trigger that. That will be something that will add to the complexity. But what in general, like for example, genomics is trying to always change like the predisposition, and all the other ones like epigenomics can be affected by the diet, can be affected, it can be like passed through generations. Even like, let's say, there are some papers of people that fast for a long time during the war, and then the generations have kept the same signature of epigenome changes at certain kind of genes. Then from what you are saying is like something more quickly, it can be at the protein level. For example, one of the things that I enjoy studying is the post-translational modification. We perform a study on high fat in a high caloric diet, and we were able to characterize the increase in the glucose uptake induced reactive oxygen species that modify the glucose transporter. Then this will be some kind of study, and this study was done with few patients, like 5 versus 5. And was actually the same patient was working as a control before the diet and after the diet, and we analyzed some tissue, adipose tissue. Then the same kind of analysis can be done, like let's say you have patients and you have them under control, let's say with medication, and as soon as you can take some samples, save these samples, and take another sample, let's say of the blood when they are during the episode, and that samples will be ideal to compare, let's say at the plasma level or at the blood level. Then actually I did have for biomarkers, you're still seeing my screen, isn't it?
Yeah.
They are different kind of ways to analyze for biomarker, and you can at the beginning identify many possible targets or genes in few samples, and in the way that you start, like that will be like the starting point for what you are asking, and then you have to confirm that using different technologies, and came out with few samples and then tested in large amount of patients. That will be a way to validate that. And what I was trying to say, like, for example, one of the important things are the sample preparation for this kind of analysis. But you just, for example, if you want to do that, I will be willing to guide you on how to prepare for this kind, depending on the question that you have. But for example, to look for biomarkers in the blood, it's important to remove this protein, that is albumin. It's like 95% of the whole protein in the blood, in order to have some samples that allow you to compare and to have meaningful data. In order to do that, you have to pre-treat the samples before do the analysis of proteomics. And by doing that, you have a wide dynamic range, and you can have identify proteins at the picograms per ml level, where the cytokines, for example, are. Then it's important to have like the experimental designs, even with few amount of samples, and then the pipeline that you are going to try for the identification of that.
Wonderful, thank you. Can I follow up? So obviously for validation you need a much larger sample, but how valid are even the information gathered from few well-controlled samples?
You have to, you do the initial, there are different approaches to do like the initial screening. You can pull them, or you can use different label techniques to analyze them individually. But then you have to also characterize or validate that specific group sample. And then you go like by steps. Let's say if you start with 10 versus 10, then you will require around hundreds, say 200s. And then in order to do that, you maybe have to require to get access to a repositorium bank, where like, let's say NIH or any of these institutions that they collect samples. And then you, let's say they identify one protein or a set of 5 protein, and you develop an ELISA, then you can go and test all of these ones. And then you start having some power analysis to establish if it's significant, if it's going to be sensitive, or if it's going to be specific.
Okay, thank you very much. Hopefully we'll have some opportunity with the other scientists to collaborate.
I will be more than happy to collaborate and to help in any way.
I have a question, Carlos. So in quite a few of your slides, you do talk about the behavior, right? So you do look at behavioral phenotype. So in the animal models, I saw that you did cognitive tests. You talked about MCI, right? So in human patients. So in some of the COPD slides, right, you were looking at, I was very fascinated by, you're looking at how these proteins are acting within a cell, right? It might be at the cell membrane and how that links to phenotype. Have you made any progress, or I know you've done a lot of work, but I don't know, have you made any progress about proteins, the activity of proteins intracellularly or at the membrane, extracellularly, and how that links to the behavioral phenotype? Or is that possible to do in your model?
We for the COPD project, we have advanced a lot, and we are ready to start publishing. And one of the things that I mentioned for the biomarkers is also true for drug development. Like you need to first secure the intellectual property in order to release the information, like at least have a chance that when the information is out there, and let's say the pharma company or you develop your own company, you have the chance of make it true. Otherwise, no one will be interested. But answering to your questions, yes, we have developed some animal models in which, like, we expose the animal to cigarette smoke and we identify the accumulation of the histone. And if we treat these animals with extracellular histone, then we can see the phenotype of the lung destruction that resembles a COPD lung patient. And if we put an antibody that blocks the histone, then this is like rescue. That in this case will not be like behavioral, but is phenotypic. It's phenotypic at the structural level. Then, but what you are saying, like, is more like at the neuronal or at the brain level. I think I saw a long time ago on a study where they identified like a small peptide and they put it intracerebral in the mice, and they recover completely from diabetes or some model that they have. Then when you are asking if you can add proteins that alter or change the behavior, it's totally doable depending of the function of the protein.
Okay. Thank you.
Fantastic talk, first of all. I want to see what your vision is. Is it the future, if you had a glass ball, is it more in biomarkers, or are biomarkers moving slowly on the backbench and multi-omics is more important to get a more comprehensive picture?
My personal opinion, they go hand to hand. Both of them evolve faster. Like the technology, the advance of the technology will allow the development of the biomarkers independently faster. And the limitation of the multi-omics is the integration part. Like for example, the example that I present here respect to COPD, it basically fulfilling the need that, or the question that we made. And it's going to be exactly the same for in your specific area. You are trying to answer one specific question, and then you will have to open the path to be able to answer that. Then they will be complementary. They are needed. The biomarkers can go like in advance, because as soon as you do each of the omics, you identify this signature of gene or proteins. And then there you have like a list of putative biomarkers. But then it requires all this long process for confirmation and validation, and it requires a lot of time and funding. And that's the difficult part. Then the biomarker is required for like the final outcome of the omics integration is trying to offer alternative drug treatments. Then the biomarker is required for the evaluation of these possible drug treatments as well. Then they have to go, for example, in our case, we were able to develop this methodology to measure the histone in the plasma. And we have some data that is ready out there that can be used as a surrogate marker for any kind of molecule that prevent the accumulation of the histone 3. Now, because we have understand which are the cells that accumulate and we have a method to make them accumulate, we can use that as a drug screening platform using our technologies and search for this molecule. Then we can go and test that in the mouse model, and we can also test in the plasma to see if, or in the bronchoalveolar lavage, if the histone 3 is circulating. But more importantly, it can be translated into the clinic, because then you can easily do that in the samples of patients to, let's say, classify the patients or to identify who are, let's say, from all the smokers who have the susceptibility to develop the disease. Or more importantly, for the ones that are identified and you start a new treatment, who is able to keep it in a low level. For example, related to the previous question also is like, we identified that this protein go high during acute exacerbation, and we can detect the increase in the protein even 1 week before the acute exacerbation to occur. Then they go hand to hand. They are complementary, but and technology is advancing, as Gladys mentioned, really fast. When I started doing proteomics, I was able to see only 200 proteins using a 2D gel, the one that you see here in the left. Then we go to like between 1,500 to 2,000 using this 1D gel. And now I am not part of the active working with the proteomics and the proteomics facility, but they implement a new methodology and have the instrumentation that allow to see between 10,000 to 15,000 proteins per run.
Okay, thank you.
Thank you, Andreas, for the question.
That was very insightful. So I was curious about your genomic and transcriptomic integration that you have shown for the COPD. So aren't you surprised you could only see one gene that overlap between the transcriptomic set and the genome? Is that something that surprised you, or?
Yeah, I personally was surprised, but then we searched in the literature, and actually even between the same kind of analysis, if you put together different kind of transcriptome analysis, they overlap only like about 30%.
Mm-hmm.
Okay. Then that's another factor, and the other thing that to keep in mind here is like the stringency that we are doing for like the thresholds to incorporate it into our study. Then that's something that for me it was like a challenge, and that's why when we have the idea of doing the interactomics, then actually we will have a higher overlap, okay, between all of them, between the proteome and all of them. And that's part of what the system biology is telling you. It's not one gene that is producing one protein. There are many genes that are like modifying or changing this gene, and they can have a different outcome.
Yeah. So in terms of like the transcriptome, were they mostly related to the, from the transcriptome that is related to the peripheral blood, or is it something that you actually got from the lung tissues or alveolar macrophages? So
that's a really, really good question, and that's important. Like in the experimental design, there are a lot of studies of transcriptome in COPD from peripheral blood, but all the omics that we have incorporated here are related to lung tissue. Okay. Yeah. Then like you saw here, maybe here in the proteome, we included our work, not only the histone 3, but the other 20 proteins that we found that were differentially expressed.
Yeah.
And all of the other ones are like the metabolome done in the lung tissue in order to have consistency. But there is another important part of what you are saying. Like I was in a meeting last year, and I was like asking for this, and they told me like the big institutions and the big consortium are doing already this single cell sequencing, which is the transcriptomics at the single cell level in large amount of populations, COPD patients versus different subtypes or different kind of controls. Then that information is coming, and that's what I'm happy, like trying to have some tools on place to be able to mix the information that I am producing to whatever is out there.
Okay.
And do not require that much effort.
From Lisa Hernandez, her question is, if I understood correctly, as a preventive measure for those individuals high risk for disease, a diet would be recommended as a preventive measure. That's the question. A diet would be recommended as a preventive measure. That's her question. And then another question, what foods does this diet consist of?
Okay, that's a really good question, and that's what I'm trying to go. As a doctor, pretty much every doctor always advise to do a different kind of diet to try to control, like losing weight is one of the major things. But in the case of Alzheimer's disease, I am very like puzzled by the glucose metabolism. And this is basically like the glucose level, like normal glycemia is below 100 at fasting. And these are like the results 5 years before the patient was diagnosed with Alzheimer's disease. Basically, for being diabetic, that is in the right part, that have higher odds of having diabetics and Alzheimer's at the same time. Basically, you have the genetic factor for diabetics, but you have also the behavioral factor, and that factor can be largely influenced by the type of diet that you are having. Then, in other words, a diabetic patient can also be controlled by having diets that are low in glucose. Then having a strict control of the glycemia in these patients will not only help to prevent the progression of the diabetes, but all the other comorbidities that are generated because of diabetes, like cardiovascular disease and neurocognitive impairment. Then that's the a recommended diet will be a diet that help you keep your glucose below the normal levels at fasting or at any time. Like nowadays, there are different ways to measure glucose. The more known is the glucometer, where you can just get one blood. But there is also like a new device that you put it like on top of your skin for a week, and that can give you in real time your glycemia, and then you can evaluate like what kind of food increase your glycemia and what kind of behaviors. Let's say when you drink or your glycemia get elevated, or when you exercise, you have to control. Then these kind of things are things that the subjects or the patient are able to control, but they are not aware. And even like subjects like me that know all these things, it's really hard to control like what you eat or stop eating or drinking what you like. Then, but that's why like the P4, we have a component that is really important, that is the supportive component. That is this one. This component is the one that said social network, consumer-driven healthcare, this component is like specialized healthcare professionals that are constantly monitoring your information. In the vision of the P4 medicine, they are going to have all your information in a database, and they say, okay, you only did 2,000 steps this week, try to go at least by 4,000 next week. There is a study that showed that if you do more than 7,000 steps per day, then you will have a longevity greater than if you do below 3,000. So then this is the kind of things that are indirectly related. But I think that's where we are going to lead by the integration of all this information. Hopefully we get there sooner than later and support it on clinical data and translational data that we can help to or contribute to generate.
She said, thank you.
Okay.
I have a quick question about the HIV work that you have done. So you have looked at the VPR to see whether there are any changes, but like I know in like the astrocytes in brain, they are one cell that actually harbor NEF protein. And is that something that you are pursuing, or in terms of changes or the setting up the inflammation in the brain?
Yeah, respect to the BPR project, I was trying to do trying to fill with another layer of information that then I am not trying to, like, my goal is not to diversify and go and search for all the protein or the viral proteins and the inflammation, but like what I have mentioned is like you develop this system. In this case, is a target cells that is overexpressing one specific protein, and that protein already induces a lot of changes that are related to the viral cycle and the capacity of the virus to hide from the drug treatments and from the immune system. Then my next step towards that project is to use that system, and now that I have characterized and I have surrogate markers of at the protein and metabolite level, to develop a phenotypic assay for drug discovery. Okay, meaning that I can in vitro, similar to what we have done here, put the macrophage that are infected and try to see which one, let's say, induce attack in one of the enzymes that got elevated, and see which molecule turn it down. Then I am not diversifying, but what you are asking, it can be done, or it can be already out there in the literature. It can be incorporated to this and try to develop a new project. But the idea is always like to get more information around that. It will help me to publish faster, or to get like consistent in, let's say, trying to get funding in a grant application, because I am keeping the same topic and I am developing around that. That's what actually I'm leading on that project.
Yeah, because I have like a NEF overexpression system that I have worked a few years ago. I have stopped working on it, but that is something because that's what I proposed earlier because of the inflammation, because there is a lot of crosstalk that happens in, at least in the animal models, as well as in the NEF protein is harbored in the astrocytes. And they, of course, it is not complete cycle, but the initial proteins are expressed. And the hypothesis is it is setting up an inflammatory system that is, of course, leads to the cognitive disorders in HIV patients.
Okay. What I can say, if you want, we can discuss more.
Sure.
And I have, for example, I am developing right now for the COPD a system to test activation of macrophage.
Okay.
Then I can measure using the same high content imaging analysis, like production of interleukin-1, interleukin-6, and TNF-alpha in response to the protein, the acetylated histone. I have developed that assay. I already proved it, and similar things can be tested in your case. Or if you, for example, want to know if the NEF protein also have some regulation in the glycolysis, then that assay is already set up. That's the part that take the most, and then you can overexpress in your system.
Sure.
And as I mentioned there, you can just extract the metabolites in a couple of easy steps, and then you send it to me, and I can analyze, and then we can answer that question.
Okay, great. Thank you.
You're welcome.